All right, part two of our video on formed elements in the blood. So erythropoietin, or EPO, is what is going to control erythropoiesis. So EPO is a hormone produced primarily in the kidneys. The liver produces just a little bit of this. And the secretion is stimulated by a decrease in blood oxygen. So that makes sense, right? If you don't have enough erythrocytes, then you may not have enough oxygen in the blood. Red marrow myeloid cells are going to respond to EPO by making more erythrocytes and releasing them into circulation. So the erythrocytes increase blood's oxygen carrying capacity naturally with hemoglobin. The increase then in the blood oxygen is going to inhibit EPO release, just like we've learned in our negative feedback system. So if you have too little, it's going to go ahead and stimulate it to release that hormone. If you have too much, it's going to inhibit it. All right, so EPO is also going to be stimulated by testosterone. That's why males are going to have a higher erythrocyte count because they have higher testosterone levels. And also that's going to affect their hematocrit, so just more blood cells, right? And the environmental factors such as altitude are going to also influence EPO levels, just because the higher up in altitude you have lower levels of oxygen. So of course that's going to stimulate EPO. And then increased erythropoiesis is going to raise, again, that blood oxygen carrying capacity and viscosity. Right? That's one of the things we talked about. You have too many red blood cells or a lot more red blood cells, you're going to have higher viscosity of blood. And if you have more plasma, then of course it's going to be a lower viscosity. All right, so this is just a visualization of how it kind of works, that stimulus of decreased blood oxygen levels coming over here, stimulating the kidneys to go ahead and release EPO into the blood, and then we're going to have that affect, right, our red bone marrow to go ahead and begin the process of erythropoiesis, and then that's going to increase the number of erythrocytes there so that we can pick up more oxygen. So just a clinical view of this, when we're talking about more oxygen carrying capacity in the body, you can use this for certain athletic events to enhance your performance. So one method of this is the self-donation of erythrocytes. So you can take away your erythrocytes, right, remove them, which is going to increase your EPO production right before a competition, and then transfuse them back in. So then you essentially just have made it so you have more red blood cells and you can carry more oxygen. The other method is using pharmaceuticals, so a chemically created EPO, and of course dangers with this is it's going to increase your blood viscosity. Your heart is going to be required to work harder, because think of it, if you've got, you know, sludge versus water, it's harder to go ahead and push that through a water hose. So same thing with your heart, and it can cause permanent cardiovascular damage because of this, and so it's banned from athletic competition. Doesn't mean that people don't still do it, but it is banned technically. So talking about the building of erythrocytes, now let's look at the other side, how they're actually gone ahead and recycled and destroyed. So again, they lack organelles, so they cannot synthesize proteins for repairs, so they do break down. So their maximum lifespan is 120 days. So not super long, about a little under half a year, right? And all the erythrocytes are going to be phagocytized in the spleen or liver. So we've talked about that before. The spleen is just the recycler of blood. Globins and membrane proteins that, of course, are going to be found inside the erythrocyte are broken into their amino acids and used by the body for protein synthesis, because amino acids, again, are just the building blocks. Iron from the hemoglobin and that is then going to be transported by the transferrin protein to the liver. And it's bound to storage proteins, ferritin and hemosiderin. Most is bound to ferritin and stored in the liver and spleen, and then transported to red bone marrow as needed for erythrocyte production, right? Because we still need that iron to go ahead and continue to make new red blood cells. So the heme group minus the iron is converted within macrophages into green pigment, which is biliverdin. It's eventually converted into a yellowish pigment called bilirubin, and it's transported by the protein albumin to the liver and becomes part of bile, which is used in our digestive system to break down fats. Bilirubin is converted to urobilinogen in the small intestine, so it may continue through the intestine or be converted by bacteria to stercobilin and then be expelled from the body as brown pigment in our feces. So you can thank the heme group from your blood cells for making your poop brown. Yes, you're welcome. It also can, of course, be absorbed back into the blood and converted to urobilin and then excreted from the kidneys as yellow pigment of urine. So, you know, your poop and pee, just thank your erythrocytes for that. All right, so we can see just this is a visualization of the body going ahead and recycling those little erythrocytes that have been broken down after a half a year of working hard, and then, you know, those proteins and the heme group and those different parts being broken down and recycled through the body or expelled. And again, it's just another visualization for us, kind of showing the different parts where the globins are going to go, where their iron is going to go, where that heme group is going to be turned into the biliverdin and then the bilirubin and then out, you know, as either urobilinogen or stercobilin or urobilin, right? And make those pretty colors. Okay. So a clinical view here for the anemia, either the percentage of the erythrocytes are going to be lower than normal or the oxygen carrying capacity is reduced. So you can have lethargy, shortness of breath, pallor, palpitations. It just depends if it's dependent on if you have too few erythrocytes or too few erythrocytes that work diligently with carrying oxygen, right? So a couple of causes can be that you have a decreased or abnormal erythrocyte formation. So something's wrong in, you know, your red bone marrow where that formation is happening. Of course, if you lose a lot of blood, this can cause anemia. Deficiency in your iron or your vitamin B12 or folic acid can happen. And again, vegetarians, vegans can struggle with that just because of not being aware of those vitamins that they need to replace that they're not getting from animal tissues. And then, of course, genetic defects. So some cases can be treated by pharmaceutical EPO, which we said that was blood doping, but in the case of not having enough erythrocytes, this makes sense. And sometimes signals are the underlying problem, right? So an undiagnosed stomach ulcer, right? You just have internal bleeding that you don't know about, which is kind of terrifying. All right, so talking about those erythrocytes, there's a couple different types of them, and I think these got a little bit swappy. So I'll start off with the antigens and the antibodies. So a person's antigen status determines their antibody status, right? So they're going to be affecting each other. So a person doesn't have antibodies for their own surface proteins, right? Because the antibodies are going to be attacking things that aren't a part of the body, right? So our antigens are like our little name tags on our cells saying, I'm Jasmine, I'm here to be in this body. And then if you go ahead and get like a transfusion from someone else that has a different name tag, then your antibodies are going to attack that, right? So it's just going to be to foreign antigens that aren't supposed to be in there. So type A blood has anti-B antibodies in its plasma. Notice, different. They're not attacking their own cells. Type B blood has anti-A antibodies in its plasma, and then type AB blood has neither of the antibodies, right? Because it's got both the antigen A and the antigen B. And then type O blood has both antibodies in its plasma, so anti-A and anti-B. All right, so here are the blood types. Got these two mixed up. So blood groups depend on the surface antigens, like we just kind of talked about, and they're projecting from the cell's membrane. And so the way we determine it, again, is if they have the A antigen or the B antigen, right? Or both of them. And they're just glycoproteins that are just hanging out on the outside saying, here I am, this is me, just little name tags. So type A blood is going to have the antigen A, type B is going to have the antigen B, type AB is going to have both of them, and type O is going to have neither of them. So you can just see their little different antigens on them, and then that O not having any. And then here's our example of the antibodies that are going to be going against those ones that aren't found in the body. So there's one other factor that affects how we determine blood type, which you've probably heard of, like I'm A positive, that's my blood type. So RH blood type is what's going to go ahead and affect that. So again, if you notice, you have the RH positive versus the RH negative. And so the surface antigen is just called surface antigen D for RH positive. And if you don't have any of that, then you would be considered RH negative. So that just determines whether it's positive or negative. So some clinical concerns about blood types, obviously, if you get the wrong transfusion, agglutination is going to occur, and that means that the recipient's antibodies are going to bind to the transfused erythrocytes and clump them together. So you're literally going to have clotting inside your blood. Of course, this is going to cause or potentially cause blockage of your blood vessels and prevent normal circulation. It can cause hemolysis, which is just the breakage of erythrocytes, and then, of course, organ damage. So not a good thing, right? Here's an example of agglutination reaction. So I'm putting type A into a type B recipient, and we go ahead and see that type B coming in, and then the A antibody is going to come there and be like, ah, and then we have agglutination there with a blocked vessel. And of course, this is just another example of a no clumping seen because they're compatible. And then we're going to go ahead and see that agglutination is going to cause that clumping and hemolysis occurring. So another interesting thing is that you can have incompatibility between a fetus and the mother. So for example, you have an Rh negative mom, so she doesn't have the D antigen on the outside of her red blood cells. It can be exposed to the Rh positive blood of her child during childbirth. So the mom now is going to have those anti-D antibodies that the baby has. And in future pregnancy, it can cross the placenta and destroy the fetus's red blood cells. So this results in hemolytic disease of the newborn. So the infant is going to have anemia and hyperbilirubinemia and heart failure. So prevention is to give the pregnant Rh negative mother special immunoglobulins. All right, so talking about leukocytes, I'm going to do that on the next video.